From Microbe TV, this is a special immune booster recorded during the Society for Leukocyte Biology conference at Michigan State University in October 2024, starring Cindy Leifer and Brianne Barker, and featuring Justin Wilson from the University of Arizona College of Medicine.
We are very excited to welcome our guest, Justin Wilson, who is an assistant professor of immunobiology at the University of Arizona College of Medicine in Tucson. So welcome.
Thank you. Thanks for having me.
We wanted to just talk to you a little bit about your journey through science and your research that you're doing right now.
Yeah, happy to share.
So how did you get first interested in science, and how did you end up on this path?
Yeah, I grew up in a very small town. I know I'm not the only one here in that realm, in the Northeast. I kind of didn't really know what I wanted to do. I just really enjoyed science and math. And I literally went to undergrad for a biology program at a small liberal arts college in Vermont called Castleton. It's called Castleton University now. And I just sort of got immersed, especially in genetics and sort of microbiology. And a very, very small section of that was immunology, which was literally a week of that class, and little did I know that would be, how I'd be spending the next 20 years of my life. But yeah, and then from that point on, I didn't have a lot of lab experience. It's a small university, so I got a technician position at a small company in Vermont that literally mined limestone that went into like paper products and different like paint fillers. And part of my job was to actually do cultures and like see what kind of treatments I could do to prevent these things from getting completely degraded when they got shipped across the country.
Cultures of what?
Bacteria, so
okay.
Soil bacteria. Yeah, so I learned how to pipette in that job. I relearned how to do dilutions, and I learned how to do like CFU counts, which ended up being really helpful, going on forward. But actually, ultimately, what it did was it helped me kind of understand. I don't want to be a lab technician. I really, really love the techniques, but I wanted to do more. And my manager at the time had got a master's degree and worked at Albany Medical College, and sort of mentioned that area. And I was like, at that time, I'm thinking like, okay. Well, I remember being in high school and just like, okay, 4 years of college. I don't want to do that. Little did I know, right? And it's like graduate school, and then there's no exact time length for that. But it sort of opened my eyes at that point of like, I really want to do more with this. I really enjoy this, but I realized it's just barely scratching the surface. So yeah, I went on to graduate school, really not knowing even still exactly what I wanted to do. I had general interest in immunology, in genetics, which I never really heavily pursued. But I joined a lab that had just gotten this big program project grant to study a bacteria called Francisella tularensis, which I had never heard of. And this was, just a couple years after 9/11, and there were a lot of CDC money for biodefense. Sure. And like, hey, we have this center-wide grant that is going to fund this bacteria that not a lot of people study because not a lot of people really care about it, but it has been mass-produced and weaponized by the United States and the former Soviet Union during the Cold War, and like a lot of places they got money to sort of explore for vaccine research. For me, we didn't often think about it in that perspective in terms of biodefense. I didn't want to feel like I was trying to like raise money, think about like the political climate at that time, but it was a bacteria we didn't know a whole lot about. And we knew it got into macrophages, which, macrophages usually engulf things, they kill them, and then, they initiate immune responses, or it's the end of the story. But this thing grew in them, and we didn't really know how or why, we knew why, but not how and what that meant. So the fact that not a lot of people were studying this was really a great project for a graduate student who anything we were going to find was going to be publishable and was sort of exciting. So it really kind of opened my eyes about the dynamic between microbes and our cells and how they sort of co-evolve to sort of either coexist or, lead to pathogenicity. And then, going on to my postdoc, and now some of those ideas have kind of carried on about, what defines a commensal versus a pathogen, and sort of how our immune system and as well as our epithelial cells sort of sometimes misdiagnose or misregulate some of those processes, and those can be disease. So maybe a commensal, but we could be treated like a pathogen and vice versa. So yeah, it's complicated, but it's a really exciting area to be in. So
what types of things did you sort of observe in those Francisella projects, and then how did that lead into things you started thinking about as a postdoc?
Yeah. So, initially what we found was, a couple things. We kind of took a lot of pages out of like the known really heavy hitter bacterial that are intracellular pathogens like tuberculosis and Legionella. So, we found that at least our project, we found a couple things. One was when these macrophages get infected with Francisella, it induces a robust cyclooxygenase 2 or COX-2 response, which leads to high levels of this prostaglandin called PGE2. And PGE2 is an important part of the inflammatory response, but like any inflammatory response, if you don't get the right inflammatory response kickstarted, then you don't have a proper, innate immune response that occurs and adaptive immune response that needs to go through there. And what we found was that this excessive PGE2 actually skewed T cell responses away from a response that would be beneficial to the host, that was almost kind of tricking the immune system, thinking, okay, behave like this when you actually need to do that. So we found that by producing this prostaglandin, which we could block to reverse, it essentially kept T cells from behaving appropriately. We followed that up and found that there's also an intrinsic effect to that PG2 back on the macrophages to downregulate a lot of the molecules, so MHC class 2, that the macrophages use to present antigen to T cells to then help initiate those responses. So shutting down both the ability of the macrophage to present to T cells as well as redirecting responses of the T cells away from a beneficial response to the host.
So I think about these things a lot where the microbe is trying to evade some immune responses, but it has to do that with control, right?
Yeah.
Because if it completely evades the immune response, it'll overgrow and then kill the host, and then it can't replicate and get passed on to another host.
Yeah.
So how is that, is there a balance there with this?
With Francisella, not so much. And I think that's what also makes it a classical pathogen, right? So especially, in this bacteria, it also depends on how it disseminates, how you're exposed to it. So, if it gets into your lung where these alveolar macrophages are, if it's not controlled, then, it can lead to death of the infected individual. Now, thankfully, if exposure to Francisella in the real world, it's highly susceptible to penicillin and streptomycin. So we used to joke about that, if we get sick, it's not that big of a deal. We weren't working with, weaponized stuff or antibiotic resistant stuff. But yeah, the point being that this is definitely not living in balance with the human host, which is probably not the case for some of the animal reservoirs, which it doesn't seem to have as much of an infection in. So sometimes we're just the unfortunate side road that these pathogens take, for both of us. No sympathy for the microbe.
So then you went into postdoc?
Yeah. And it's funny because I applied to all these labs that were doing very kind of hardcore host pathogen interactions. All these people that trained under Stanley Falco, and people that were doing like Legionella work, TB work. And then some, and I also applied to another lab in the University of North Carolina at Chapel Hill, Jenny Ting's lab, because at the time she had some papers with tuberculosis, and I knew her from her inflammasome work, and actually invited her to come give a talk when I was a grad student. And I had a really good interaction with her. So she was sort of the one lab that wasn't as much in the same vein, but that was the lab I ended up joining. So that really kind of redirected my work towards other areas, which is kind of where I am now. So in that lab, the link between what I just described in here, it has to do with the microbiome, right? So like a lot of things do. And at the time, the lab was just starting to get into looking at gut inflammation, both in the context of inflammatory bowel disease, cancer, and sort of general inflammation. And the lab studies this group of proteins that are known as the NLRs, the nucleotide binding leucine-rich repeat proteins, that are these intracellular sensors that see components of microbes, PAMPs, or things that are released from cell death, like if you slip and fracture your foot, all the things that come out of the injury, right? So these are the sensors that see those signals and say, okay, something is wrong here. And they have this sort of multi-protein complex that assembles, and part of that is just for the tight regulation that has to occur in order to get those inflammatory cytokines out because of their potency. So we were really interested in looking at how both the microbiota, facilitates signals through those sensors to then lead to inflammatory responses in the gut. Because some of these sensors have been implicated in Crohn's disease, which is still some of the most largest links between these sensors. Yeah.
And there's a link, right?
Yes.
There's a link, yeah.
You guys knew each other when you were postdocs.
Yeah, so we were both postdocs together. And I was actually going to ask you sort of what particular sensors were you really interested in, and sort of how did that develop into things you're working on. But I was going to ask that because I kind of knew some parts of it because I might be on at least one of those papers.
So there's two different genes that we worked on, and I think it's common for a lot of postdocs. Okay, here's a project for you that should be going in a direction, and then you're also sort of supposed to develop your own project. And if you have faculty, academic mindset, then you got to keep thinking about the whole time, which one of these can I take with me? That way I can actually, build a career out of it. So one of them had to do with this protein called NLRP12. So there's a bunch of these NLRs. NLRP12 is sort of this weird one that it doesn't form an inflammasome. There's some reports that it does, so it may depend on the context. But what we really had shown and others had shown was that it's a potent inhibitor of NF-kappa B. And it sort of drew me a lot to this sort of the same idea of like with the Francisella story. I've always sort of been interested in what are the negative regulators, what are the things that sort of shut processes off or skew them in a different direction. So I think that was sort of attracted to that. But what we found was that this protein, at least in the intestine, if you get rid of it, you no longer have a normal ability to suppress NF-kappa B, which is the master transcription factor for most of your pro-inflammatory cytokines. So you have excessive inflammation, and they have these animals that you delete this gene in. If you put them through models of intestinal inflammation and inflammation-driven cancer, they get more inflammatory bowel disease and more cancer. What we also found was that the microbiome that was harbored in these mice actually changed. And what was sort of intriguing was that at least what we thought was happening was the inflammation that was increased as a result of loss of this protein led to sort of shaping of the microbiota. As we know, inflammation can impact the microbiota just like the microbiota can impact inflammation, which makes it complicated but also exciting. And what would happen was we found that there was a depletion of this group of bacteria called these Lactobacillus. And we found this was strikingly low in these mice, and we collaborated with Vincent Young at the University of Michigan, who was also defining a number of these Lactobacillus. He was kind enough to send us a cohort of these bacteria that we could put back into the mice. And what was really exciting was that the mice had still like low levels of inflammation just because of the nature of missing that gene, but they were largely less susceptible to the gut inflammation versus the mice that were just given untreated or given just like the broth that you grow the bacteria in. So if you replenish these microbes, put them back in, it could help offset a lot of the inflammation that was occurring in the gut.
So even though it was really important for the inflammation, it's doing that through this loss of the particular type of bacteria?
I would say it's both. Yeah. So we could take cells from these mice or delete the gene in cell culture, and you see this elevated inflammation. So we know that mechanistically, like on the molecular level, that you lose this protein, you have a heightened response to inflammation and a baseline increase. That translates in the animals, but they also have this aspect of the bacteria there. If there's more inflammation in the environment, that's going to change the microbes. There's tons of factors that the gut produces from mucins, antimicrobial peptides. Gap junctions change, which allows movement of these bacteria into the intestine. All those things are probably impacting this, and that over time changed. Because the mice were born, you're sort of born sterile-ish, and then you get populated and your microbiome, progresses as you go from, consuming milk to whole food and so on. So, we could track some of these changes, and then putting the bacteria back in actually seemed to help with the disease. But, we never did this experiment, but I'd be amazed if that bacteria didn't revert to the original, loss of these microbes if you let them continue on, just by nature of having this gene missing, right? It's not able to completely turn off that inflammation because those microbes are there, they're triggering inflammatory responses, but we have, regulatory mechanisms to kind of keep that in check, and this gene was part of that and it wasn't there anymore. So yeah, multi-steps, but it helped us kind of understand, some of the steps of how these sensors sort of respond to microbes and then shape them and vice versa.
So you mentioned there were two sensors.
Yeah. The other one, which has become my kind of personal favorite, is called AIM2 or Abstinent Melanoma 2. So this is a double-stranded DNA sensor. It's expressed highly in immune cells, but one thing we discovered, and others had showed this around the same time, is that epithelial cells also express this gene. And it sees double-stranded DNA, and kind of the long story is abstinent melanoma. It was found in a screen of melanoma cell lines and some tissue, but very little research has actually been done in melanoma. Several groups through Germany and a few others have shown that this is also a gene that's lost in a number of colon cancer cases. And it wasn't really known why, and at the same time, a group of immunologists did a forward screen and found that AIM2 has a lot of the same structure as all these other NLRs. It forms an inflammasome, but it has this DNA binding domain instead. And they went on to show, as a lot of immunologists would, that this is important for double-stranded DNA viruses. This is important for intracellular bacteria like Francisella tularensis, which I did not make those links, and that would have been really cool, but it was sort of ironic that those two things kind of came together. So it's great for intracellular bacteria and certain double-stranded DNA viruses that haven't found ways to overcome it. But then we were interested in the context of cancer. Again, there was that clinical link for the gut. We knew that the inflammasome was important for gut inflammation, so we thought, well, maybe it's inflammasome. And what we found after a very long time was it wasn't inflammasome that was doing it, but AIM2 did suppress colon cancer. And what we discovered, it was sort of this inflammasome independent role for AIM2 through suppressing the PI3 kinase AKT cell survival pathway, which is a pathway that's very involved in a lot of cancers. So if you mutate this, you often have dysregulated growth, and that's one of the many components of what gives rise to cancer. Yeah.
So then how did you take that into what you're working on in your lab now?
So this is sort of all, it's a bit convoluted, but sometimes you follow the data, and that's where it takes you. So that very question I took with me to my lab at the University of Arizona. I started about middle of 2018. Got the lab set up a year or so in, and then actually saw you the day before the world shut down.
That is correct.
But at that time, we basically had the question of, all right, going back to that idea, let's take a step back. AIM2 is a sensor of microbes. It also it can sense self-DNA like mitochondrial DNA or nuclear DNA, so it can be a DAMP sensor or a PAMP sensor, which is great when you're trying to understand inflammation, both intrinsically and from a microbe point of view. These pathways, these PI3 kinase pathways, these inflammatory pathways, they're very important for cancer, but the reality is your gut has to deal with these insults day-to-day basis, whether it's what you're eating, you're interacting with microbes. You got to get gut homeostasis right at day one, or you're never going to live long enough to see cancer, right? So we really just sort of had that question of, okay, if this senses these microbes, or if it facilitates these important homeostatic responses, what does that do to the epithelial cells of the gut, which we know also express AIM2, and that's what we found was playing the role in cancer, just during normal homeostasis and during different inflammatory insults. So we set up one of the first things we did when we came in, I got a really talented master's student who was really excited about gut inflammation. She's like, I've never had any. She told me she was really worried about working in cell culture. And I was like, I got the perfect project for you, organoids, right?
Oh,
right. So, talk about let's start simple and go extremely difficult. But she took to them really easily. She actually defined a lot of the systems that we were still working with now. But essentially what we did is we take epithelial cells from the mice, we can isolate them from the tissue, grow them up in 3D matrigel cultures, and then just supply different growth factors to keep them either in sort of stem-enriched states or more defined differentiated mini guts. So we developed this platform with collaborations. We did a little bit when I was at UNC, but we sort of had to refine it in my current lab. And we deleted AIM2, and we basically said, okay, well, what if we look at all the different markers that are associated with epithelial cell differentiation? And I always have to kind of remind the immunologist, the gut epithelium isn't like how we like to picture it in the little images. It's not all the same cell, right? You have goblet cells, you got cells, which make antimicrobial peptides, you got enzyme-producing cells, and so on. But you also have these other cells that are really emerging called tough cells. And I didn't really know much about these until going to some conferences and including this in our screen. And what we found, and we haven't published this yet, we're getting it submitted soon, is that when you deleted AIM2, we saw a loss of these tough cells in these organoids. And we really just kind of pursued it. What's really cool is that intestinal epithelial cells, if you hit them with IL-4, IL-13, these Th2 cytokines we usually just think about, in terms of T cells and IL-Cs, that reprograms epithelial cells to actually make goblet cells and tough cells. And we could see a similar defect in the AIM2 knockouts when we pushed with IL-4. Tough cells are really exciting. They're these low population epithelial cells, as long as you don't have a rapid worm infection. So it's thought to be maybe about 1 to 0.5% of your total epithelium. But if you get in contact with a helminth, parasitic worm infection, you get this massive, robust, tenfold induction of these tough cells. tough cells have different receptors. One of the ones we know that they use to sense is a receptor called succinate receptor. So, we all make succinate, right, in our TCA cycle, but it usually stays in the cell. Certain microbes, a lot of microbes in the gut microbiota, as well as a lot of protozoans, make high levels of exogenous succinate. And tough cells will see this, make high levels of this protein called IL-25, which activates a type of immune cell called the type 2 innate lymphoid cell, or ILC2, which makes the IL-13 that tells the intestinal stem cell, okay, make more. And it's a sort of weep and sweep idea that you get a lot of fluid loss, you get muscle contraction. It's basically like, let's get rid of the worm. Yeah. And a couple of groups have showed this. Jakob Moltke's group, who's now at the University of Washington, several others, really define this. And since then, people have said, okay, this isn't just involved in helminth infection. There's important implications in inflammatory bowel disease. So Ken Lau out of Vanderbilt a couple of years ago now showed if you push these tough cells, you could actually offset mouse models of inflammatory bowel disease. Apparently, norovirus loves to infect these cells.
I was thinking that, yeah.
So if you get rid of tough cells, you don't get norovirus infection. There's been a couple papers with tumor immunity in the gut, just because, kind of like the Francel story, if you skew the immune response away from a way that it needs to help, in this case, usually you think about like a Th1 or Th17 response, and for good anti-tumor immunity, you can kind of shut down those responses. So our understanding of this is really growing well beyond, and we never had any interest in getting into helminths and anything, and we might at some point, but we have a lot of interest in how these cells sense microbes, because those worms, they don't use succinate. You can delete that succinate receptor and the worms still impact tough cells. We have a lot of interest in how the microbiome actually impacts those and what that does to gut homeostasis during different immune responses and during different insults.
Have you looked at any differences between commensals and pathogens, say?
Oh, yeah, we're just now getting set up with some Citrobacter models in our lab. We've done a little bit with germ-free mice and doing colonization of like normal microflora, and we've seen some differences in tough cell induction with normal gut microflora reconstitution, but we haven't gone too far down the pathogen road. A big part of this past year has been sort of rebuilding the lab and getting more people in. So a lot of the time has been getting really good people kind of up and running to ask those questions. And the organoid work takes a lot. Takes a lot.
I can imagine.
Yeah.
I was wondering if you could, before we wrap up, just say something quickly about SLB. Like, what is your interactions with SLB, and why are you a member?
Yeah, well, this is actually my first leukocyte biology meeting, but I've gone to American Association of Immunology, AAI conferences every couple of years, and I always remember, even from graduate school, that I always really liked the SLB sections. I always made a point to try to go to those. So it's surprising I hadn't gone to one of these before. And I sort of decided, okay, this is going to be the year, we'll do it. The other thing I'll mention real briefly is we have another story that I'm going to talk about for the first time today too, which is our lab actually started developing into the oral cavity to ask all the same questions about how the microbes and the immune system in the oral cavity initiate either beneficial or detrimental responses, because a lot of that biology isn't really well defined. And we actually published earlier this year by starting to look at different oral streptococcus species that we know are present in the human oral cavity, but some are linked to cancer, some inflammatory links that may indicate what dictates more pro-cancer associated versus sort of commensals. So this seemed like to be a really good platform to kind of talk about that story and some of our follow-up work, which, as you can imagine, has inflammasome implications.
Of course.
But yeah, more of it. We're still working on that.
Of course.
But,
so before we wrap up, do you have any advice for trainees?
Oy, how much time do we have? Yeah, I think there's a lot of things, right? I think you gotta do something that you're passionate about. I mean, this is not an easy job, and I think so much of this should be like, you should really have time and really think about like what you want to do in the area that you want to work in, and find a project that just really kind of grips you and keeps you up at night for good reasons, that keeps allowing you to ask questions, and have a solid a group of people that you can talk to, both about science and about random stuff, because you're going to be there for each other. You're not the only one that's going through these kind of things.
Make sure you have colleagues you could drink beer with, say.
Exactly. Yeah, or whatever it is. Cycling or playing pickleball, whatever the cool kids are doing these days. But find something you're passionate about. Have some good outlets to kind of take your mind off, and also to refresh your mind, because then you'll come back with very much a new way of thought, and then get out there. Go to these kind of seminars, go to conferences, because you never know. You may see a talk on something that you think is completely unrelated, and you're like, hey, that explains what I'm looking at right now. And plus you get to meet collaborators, and I've had so many good interactions with collaborators. Like that's been such a fun part, and it's great post-COVID world of being able to do this again.
Yeah.
Yeah.
That's awesome.
Great.
Well, thank you so much.
Yeah,
Thank you.
thanks for having me. This has been great.
Awesome to have you. And really enjoyed talking with you.
Yeah, really enjoyed that.
Yeah, anytime.
Very exciting.
That's a special episode of Immune and Immune Booster. Questions and comments, please send them to immune@microbe.tv. And if you enjoy our work, please support the science shows of Microbe TV at microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today from the University of Arizona College of Medicine, Justin Wilson. Cindy Leifer is at Cornell University, Cindy Leifer on Blue Sky. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious.